Thank you. Thank you so much, Tatiana. Should I share my slides now?
Yes, please go ahead. Feel free.
Okay. Hi, everyone. I hope everyone can see my slides. Maybe
Yeah.
a thumbs up okay, lovely. So, thank you, everyone. It's my great pleasure to be here and to be giving this boundary-breaking science seminar today. I heard that is one of the first ones in this format, so very, very honored. Thank you to all the organizers for making this possible. I'm Na Tai. I'm a assistant professor at ETH Zurich. As Tatiana said, I just moved here 2 months ago. Prior to that, I was in Helmholtz Munich, where most of this work that I'm going to be talking about is done. So today I'm going to talk to you about genetics of major depressive disorder, or what we normally in daily life call depression. So I'm a statistical geneticist, and what that means is I use statistical methods to test for effects of genetic variations on phenotypes. And these phenotypes can be things like traits like height and weight, or diseases such as diabetes or depression. And usually for these tests to work very well, we need genotypes on a large number of people and good phenotypes on the same people where we have genotypes on. And so I want to spend like 2 minutes talking about what good means. So good could mean a lot of things. A good phenotype could be one where we don't currently understand very well, but we really need to understand because there is a lot of social good coming out of studying it, or also because, it's a very challenging phenotype to study. So in this sense, depression or major depressive disorder is the perfect phenotype. Definitely fulfills the criteria of good social, outcome if we studied it. It is a very common disorder. About 20% of the population would have it at one point in their lives. Currently, we don't understand it enough to be able to diagnose it or treat it effectively. So there is a great need to understand this disorder better in order to treat it more targetedly, so as to make, lives of people a lot better. And at the same time, it is extremely challenging to study a mental state. It might even be the greatest puzzle there is to understand why we have certain moods and why it persists. So in this sense, depression is a really great phenotype to be studying because, it's important and it is very intellectually challenging for academics. The problem is good could mean a whole bunch of other things, such as, it needs to be reliably and well measured. We really appreciate low measurement noise in phenotypes that we study, and etc. So in this sense, height is a very good example of a good phenotype because I can very reliably measure height, adult height, with any form of decent tape measure. And it would be the same if I did it today or tomorrow. And it's very low contribution from environmental factors. And it is the result of biological pathways controlling bone growth and stuff like that. So in that sense, depression is a terrible phenotype. This is currently on the slide, the current diagnostic criteria for depression. And as you can see, it's made up of 9 symptom criteria, along with other criteria not shown on the slide. And so basically it's a laundry list. If you fulfill 5 out of 9 and, out of which 2 needs to be either of the first 2, then you're considered clinically depression. However, none of these symptoms specifically map onto a set of known biological mechanisms. This is not even an exhaustive list. There are many other scales used to assess depression that contain other symptoms. And and sometimes these contain contradictory things, such as increase or decrease in weight or appetite. Very rarely do you see for a disease that you can have either extremes map onto the same disease. So terrible, terrible phenotyping. In other words, these symptoms are best seen as indicators of an underlying disease whose nature is yet to be discovered. And this brings us back to the challenge of discovering the nature of depression, which has been named, or depression or psychiatric disorders in general, which has been named the main task of psychiatry 12 years ago by Karl Jaspers. And there's a long history of psychiatry, much of which happened here in Germany. And what I can tell you today is that work over the past 10 years or so hopefully is bringing us closer to knowing what's going on in psychiatry out of 110 years of history. So 10-ish years ago, I was a second-year PhD student in a group in Oxford of Jonathan Flint's, where I was learning to perform genome-wide association studies, or GWAS, on depression. So back then, GWASs are usually done on case-control data sets where we collect a large number of people with depression, we call depression cases, and a large number of people who don't have depression, who we call controls. And we would get genotypes and millions of their genetic variants in the genome. We would then test each genetic variant for association with the disease. The result of a GWAS is usually visualized in the Manhattan plot as shown on this slide. On the x-axis, you have the position along the genome, and on the y-axis, you have the negative log 10 p-value of the association test. Each dot represents the association result of a single genetic variant with the disease of interest, and those above this dotted red line are considered statistically significant, and those are considered hits. So what you mean when you say, I want to find the genes of a disease is to perform a GWAS and have genetic variants on the, near a gene, and you call that a gene for the disease. So the first demonstration that GWAS as a method works was in this paper published in 2007, where GWAS hits was found for a variety of diseases, including hypertension, rheumatoid arthritis, diabetes, etc. And this is using data from thousands of cases of each disease and more than 10,000 shared controls. So our study was published in 2015, and it was the first study that has found the first 2 genome-wide significant hits for depression. And when I first got these results, I was, happy that, I was going to graduate from my PhD. And my supervisor was over the moon and said, you realize this is a big deal because it is the first time that genetic variants has been linked to depression. Now, in the subsequent years, we found several more interesting things using this data, which is 6,000 severe depression cases and 6,000 controls. For example, we know that stressful life events is a big risk factor for depression. So we wondered if those people who get depression without having had a very stressful life event were more at risk because of genetic factors. So what we did was we removed everybody in our 6,000 cases, 6,000 controls data set who had severe stressful life events, leaving us with only 9,000 people in total, 4,000-ish with depression and 4,000 without. We reperformed the GWAS, and we found 3 different hits from what we got in the full sample. And later on, my collaborators and I then developed statistical methods to test for interaction effects between genetic variants and environmental factors like stress, and showed that these 3 genetic variants, which we found only in the non-stress individuals, interacts significantly with stress, while the original 2 hits that we got did not interact with stress. So this leads us to a new form of analysis that we are able to use to identify the interactions between genetic variants and environmental factors towards disease. While we were playing with our the same data sets, the world and data that is being collected to do this kind of analysis was changing also. In particular, what happened around 2015 and 2017 was the dawn of the era of large publicly available data sets. It started with GTAGs, which is a resource of gene expression data in more than 40 tissues assessed in hundreds of individuals, and this was released in 2015. But really what galvanized and really transformed genetics, in the genetics community was UK Biobank, which became available in 2017. And this is a data set of 500,000 individuals, genotyped and phenotyped, and released for research purposes. So back in 2017, it really felt like the race is on in terms of using this data set to find things in terms of genetic associations with diseases. So in the subsequent years, GWAS on depression, just like GWAS on other diseases, became much, much bigger than the 6,000 cases and 6,000 control setup that we had before. With the increasing available of large-scale biobanks and datasets from consumer genomics companies like 23andMe, more and more hits were found. And as you can see, in subsequent years, many, many more GWAS studies were performed. And we are now up to not 102 significant locus. This is a study done in 2019. We're now up to 600 hits for depression. So all that seems wonderful, and it's like amazing amount of progress in just a few years. But there is one worrying caveat, and that is these studies seem to have assessed depression through asking different questions. So some studies have asked, have you been treated for depression? Some studies have used depression codes identified from medical records. The combination of all of these different types of phenotyping has resulted in these massive GWASs. So then the question that I had, along with some other collaborators of mine, seeing that, all these different types of definitions were used in performing GWAS, is whether they're all the same as clinical diagnosis, which we had used previously in case control cohorts, and whether they should all be interchangeably used in a GWAS setting. So I performed an analysis a few years back using data from UK Biobank, where there's many different forms of depression diagnosis, ranging from clinical diagnosis to self-reported diagnosis. And what is evident in this plot, which shows the definitions of depression on the x-axis and the total genetic contribution to each of these definitions on the y-axis, is that self-reported depression, shown in green, has a much lower genetic contribution than clinically defined depression, as shown in purple. So what this means is that if we combine the green and the purple definitions of depression in a meta-analysis, as we've seen in many of these studies shown on the earlier slide, we will end up with a heterogeneous hybrid, which would probably give us heterogeneous GWAS findings that may not be specific to depression. So then now we are faced with a dilemma, because we have a lot of data here on these types of definitions, but not so much on these types of definitions. So should we throw away all of these in favor of all of these things, and never make use of the hundreds and thousands of data we have on people? So obviously not. My collaborators and I, we really want to find a way to extract as much information as possible from what we call shallow data, these sort of self-reported data that may not be clinical grade, but we do want to make the best use of them. So this is how normally data looks like in biobanks nowadays. We have very few individuals who've answered questions on what we call deep phenotyping, or the sort of strict clinical criteria phenotyping for depression, but a lot of people who have answered, shallow depression phenotypes, such as have you had depression before, self-reports, etc. These people have also normally also answer questions on other mental health measures, their socioeconomic status, their lifestyles, their sort of, status on other diseases, etc. So what we've started working on a few years back is imputation, which means to fill in the missing values of this clinical grade depression using data from all of these other phenotypes that we have on these individuals. And we can perform imputations in several different ways. We can use previously published statistical approaches, for example, this one here, shown on the slide, based on singular value decomposition. We could also do this by developing novel deep learning methods for this approach. And our results are quite promising. We are able to get more GWAS hits using the imputed data from either the statistical-based approach or the deep learning-based approach. And we are able to show that the results are also, not only do we get more of those GWAS findings, they are also specific to depression. So in other words, we don't compromise on specificity while we increase the power. I don't have time to show you all of the results on these today, but if you're interested, please do get in touch with me later on. I'll take the last few minutes to chat about some ongoing work in my lab that I'm very excited about, as well as some future work that I think will be important to bring our research closer to applications in the clinic, for example, in terms of drug design. So far, I've talked about different definitions of depression, but what we know is that depression itself, even when defined with the most stringent clinical criteria, is still very, very heterogeneous. It could be in terms of its clinical presentation, in terms of the kind of symptoms that different patients come in with, or also in terms of onset. So different people have depression at different points in time in their lives, and that's clinically significant. In fact, a lot of genetic evidence also suggests depression is the common consequence of different biological pathways. To illustrate this problem of heterogeneity, I have a cartoon here. So let's say depression, there are two types of depression, and one type is illustrated with this orange bit, and the other type is illustrated with this blue bit. And this is just shown very abstractly using the circle over here. The task that we have at hand, not knowing where the distinction between orange and blue is we want to be able to separate subtype 1 from subtype 2 and identify what are the genes that are involved in this orange bit versus what are those that are involved in this blue bit. We don't want a situation where the subtypes we identify are this way, where we do see differences between this bit and this bit, but they don't align with the biological pathways. So this is a challenging task. And one student in my group is currently working on methods that would allow us to distinguish between these two situations using genetics. And we hope that this project will come to fruition soon. In order to get sufficient data to fuel this efforts, I've started a heterogeneity focus group within the Psychiatric Genomics Consortium, which is an international consortium of researchers that work on psychiatric diseases. The goal there is to amass from all global cohorts symptoms and clinical criteria and clinical features of depression, and to characterize each of them genetically in order to identify those with genetic relevance to depression and to use those to subtype depression. So this is all ongoing work. Secondly, we really want to push sort of the molecular side of things in terms of understanding depression. And what we know is that stress causes depression also, in addition to genetics. In fact, many times stress is a bigger risk factor to depression than genetics is. So what we've done is we've now started looking at how stress changes gene expression in the brain in rodent models. And here is actual research figures that we have from a research study we've done on mice subject to chronic social defeat and controls. And here the little dots are just individual cells in the brain that light up when we subject mice to stress versus when we don't. So we're making these cells light up using a genetically engineered approach. So we subject mice to 4 weeks of chronic social defeat, and we use this fluorescent labeling method in a genetically engineered mice to figure out which areas of the brain respond the most to chronic social defeat and which areas don't respond that much. And once we've identified those areas, we then perform spatial and single-cell transcriptomics on slices of the brain that covers most of these identified stressed regions to then identify which genes contribute most to these gene expression differences. Part of the work is to develop methods to analyze this data. So we're working with collaborators to develop this Bayesian machine learning model that identify regions of the brain that show significant gene expression differences based on spatial transcriptomics of the stress and non-stress mice, so as to narrow down our search for stress-responsive neurons and the genes that are differentially expressing them. So I've come to the end of my talk, my side of the talk, and I've covered how genetics of depression has come a long ways. We have current developments on how to maximize the use of data from biobanks and EHRs, especially with regards to self-reported data and non-clinical grade shallow data. And I've also talked about our further investigations into depression heterogeneity and the molecular basis of depression using mouse stress models. And there's obviously a lot more to do in terms of, bridging our efforts with efforts in drug companies like Bayer to actually bring these research findings to clinical translation. I would like to thank all of my colleagues, my friends and family for supporting some of these research, and also my funding sources and all of these people who have given their personal data for research purposes in the UK Biobank, iPsych and psychiatry genomics cohorts. And I'll hand over to Martin, who will be able to tell us the drug development side of things, and what we hope for in terms of being able to make these genetic findings available as translational options later on. Thank you very much for listening to me.
Yeah, thank you very much, Na, and thanks a lot to the Bayer Foundation for inviting me. Just a kind of a quick introduction of myself. I've also been kind of involved in neurogenetics a lot during my career. So I've started with genetics of language disorder, then moved to the genetics of autism spectrum disorder, where I kind of got very familiar with brain tissue and kind of developmental genetics. Now with Bayer, I'm working on the genetics of cardiovascular, renal, and immune system diseases, and that is kind of obviously in a very different population, so more adult and older individuals, and the tissue is all over the human body. So those are quite different experiences. And especially in Bayer, I'm working in the kind of identification of suitable targets for kind of any kind of indication in the cardiovascular and renal space. And AstraZeneca quite nicely has published a paper in 2018 already, where they stressed 5 R's. So in order to kind of develop a successful drug, one needs to take the right target, right tissue, right safety, right patient, and the right commercial potential into account. And I think it's quite obvious. I mean, now the right target is pretty much put to the left side here, that the kind of decision of the right target needs to come quite early in this whole kind of process. So there's a lot of burden into identifying the right target early on. And of course, right can mean a lot of different things, and it's quite difficult to kind of really make a good decision here. Well, luckily we have genetics, and we have quite generally saying that genetic evidence increases kind of the odds of a gene to eventually making it an FDA-approved drug. So in this table here, you can see a lot of successful drug targets that do have a genetic association to the indication that they eventually kind of are used to treat. And this table is just not just an anecdotal kind of story, but very systematically. A lot of academic groups have published findings to show that indeed genes overall that have a genetic association with an indication are going to be more likely to make it at the end towards FDA approval, as well as to all the kind of clinical phases in between. Now, Na has quite nicely introduced the kind of the use of the idea of genetics going from a phenotype to a trait, and having that is of course amazing. It gives us a justification to kind of use that target and move it forward, and to kind of spend budget on it. However, we still need to mind the gap. There's still a lot of things to fill in between, as kind of Na has shown with single-cell RNA sequencing. We need to make sure that we are then that this drug target or this gene is actually expressed in a tissue where we would expect it, and that it's modulated in the way we would expect it, or at least find out how it is. So in my kind of daily work then, when kind of identifying a good or a right target from genetics data, I can face kind of different situations or use cases. And the ideal use case is that for any indication that where we want to find a new target, we have a very powerful GWAS with maybe hundreds of genes being associated to the disease in question. And that would make our life a lot easier. That would be a luxurious situation, because then we can, from those 100 genes, select the ones which are most easy to validate because we have the cell type already in place as a simple assay. The target might be easy to drug because it is an enzyme or a receptor instead of a transcription factor, which is more tricky. Ideally, also, there might be kind of a good body of literature that further validates that this gene is actually doing something in the indication. And then next to a plus that there is like no or low competition. Right? The more genes you have with an association, the more fine-grained you can be and cherry picking you can be. However, then once you have picked the gene, there are still a lot of things to be filled in. So, for example, the timing. So when does this gene actually contribute to the disease, and then which tissue and cell type does it contribute? So that we can really build a kind of thorough pipeline and with a proper information on the mode of action of this gene, how it's contributing, when and where should we intervene. However, in a lot of cases, there's the other use case where we don't have any GWAS signal, or maybe just very low GWAS signal, and there's 1 or 2 genes that might be associated, and then we don't have this luxurious situation where we can just cherry pick. And then in the case of 0 genes being associated, what we can do is either wait for GWAS to become more powerful, and as Na has showed for major depressive disorder, they have improved the signal quite a lot over the years. Or again, as Na was showing, we can use kind of related phenotypes. Either we can kind of pool a lot of subjects in order to increase the sample size, and then again, we would increase heterogeneity and maybe lose some of the signal, or we can build kind of smarter phenotypes in order to decrease the heterogeneity. So those kind of 3 different approaches are things that we are doing. However, then in addition, we would then have to further increase our hypotheses and then really be able to kind of show that this other phenotype that we have built is actually relevant for the indication that we want to treat at the end. So there's, of course, well, I call it the third use case, and that is all the FDA-approved drugs that don't have a genetic signal. So I've earlier shown you that a genetic association does increase the odds ratio of FDA approval eventually. However, still, like more than 80% of the FDA-approved drugs do not have a genetic association to the indication that they're being used to treat. So here's the kind of one problem is that, well, one reason that this of this gap of the 80% of genes could be that the current level of genetics, the current genetics data is just not sufficient. So it doesn't have reached saturation level. So there's still a lot of kind of value in pushing genetics and kind of increasing sample sizes, improving the phenotyping, so that eventually we can see more and more of the FDA-approved drugs to have the genetic association, and then also for us kind of making our life easier by being able to pick those targets which are easy to drug. And on the other side, it's going to be really valuable to evaluate in such a systematic manner, not only the genetics part, but all the other kind of omics layer or kind of genome-wide layers that are available, such as the transcriptomics, single-cell transcriptomics, proteomics, and so forth, in order to really find out how do white targets look at all the other kind of omics layers. And there was this example of a preprint from Dennett in 2024, where they have, for example, showed that a target gene, which is very cell type specific, does have a better chance of reaching the different clinical phases. So this is certainly a way to move forward, and being able to early on say, how does this property or characteristic of a gene may eventually influence how successful this kind of pipeline project will be at the end. So again, to say what it is really important to kind of pick the right target. Oh, sorry, my daughter just came in. It is, well, it's first of all, it's really important because picking the right target early on will help us reduce the attrition rate by picking the right targets, and it will improve the pipeline activities by having a stronger hypothesis from the get-go, so that we don't need to test 10 different cell lines, but we know exactly which cell line and which cell type we should investigate. So as a summary, similar to what Na has said, I think genetics is really a strong predictor for pharmaceutical success. It is not that easy to get GWAS hits, and it's more, it's more difficult to get the right target. And there's much more to do, and we are certainly on a good way. So finally, thanks again to the Bayer Foundation. Tobias Strunz, who helped me last time I had to present with the Bayer Foundation, and he quickly jumped in. Everyone from my team at Translational Science, and people that I'm working with, and the Bayer team. Thank you.